Já pode começar.
Ok.
Hello, everyone. So thank you, everybody, for the ones that are here and for ones that are online. So I have the pleasure to introduce to you today Dr. Jefferson Antônio Leite. He graduated in Biological Sciences at the Centro Universitário de Faculdades Integradas de Ourinhos in 2016, and follow up with his master's under the supervision of Drs. Daniela Carlos and João Santana, and then with his PhD supervised by Dr. Niels Olsen and Daniela Carlos, in basic and applied immunology at Ribeirão Preto's Medical School in USP. During his PhD, he had the opportunity of doing a joint work at the Institute for Molecular Medicine at the University of Medical Center Johannes Gutenberg University of Mainz in Germany, where he went on and then all of that during his PhD. After that, he continued for a short postdoc at Dr. Niels' lab, and then went back to Germany, where he spent 2 years of his postdoctoral training over there. He then came back, and since last year, he's become assistant professor at the Microbiology Institute in the Immunology Department and here at UFRJ, being the PI of the T lymphocyte biology laboratory. He has won many awards and prizes, being that Teresa Kipnis, one of them, that is presented at our annual meeting at the Society of Brazilian Immunology. So over here, he has dedicated his work, as he will be telling us, on T cell function. And he's also doing some people resources training with students at PhD, master's, and undergraduate students in the lab. He's also the International Affairs Deputy of the Immunology and the Microbiology Institute. So Jefferson, it's a really great pleasure to have you here. Thank you again for accepting our invitation.
Olá, pessoal. Seguro. Thank you, Miriam. Thank you, Bruno, for the invitation. It's really a pleasure to present in this webinar. And today I'm going to talk about some of the research that I've been doing the past few years. Most of this data that I'm going to show is data from my postdoc time that I did, as Miriam mentioned, during my postdoc in São Paulo and later in Germany. And it's about my favorite cell, T cells. And I'm going to talk about specifically Pois? Será que tem que ficar mais perto lá? Não. Tá. So as I mentioned, I'm going to talk about T cells, and I think I don't need to give a deep introduction about T cells to this audience, as we all are immunologists here. So we know that T cells originate from progenitors in the bone marrow, and then they go to the thymus, where they go through several maturation steps, and then after these steps, they become mature CD4 and CD8 cells, and they go to the secondary lymphoid organs, where they are exposed to antigens, and they do this through the antigen-presenting cells. As you can see here in this nice video, in red, you can see the T cells, and in green, the dendritic cells. And the DCs are constantly presenting antigens to T cells in the lymph nodes, for example. And once the the T cells recognize an antigen for which they are specific. They become activated. So once they recognize an antigen for which they are specific, they become activated, and these require, as you know, two signals. The first is the recognition of the antigens through the TCR, and the second signal is the co-stimulation. And these will drive the activation of the cells that will give signs of survival and proliferation, and also depend on the immune response that is being generated, like depend on the pathogen, for example, the antigen, the dendritic cells release different cytokines, and these cytokines will determine the fate of these T cells. And there's different subsets of T cells. In particular, we are more interested in the T helper compartment. And there's, like you can see here, different T helper cells like Th1, follicular T helper cells, Th2, Th17, and regulatory T cells. And I'm more interested in the past few years in Th17 and regulatory T cells, which is I'm going to focus more during my presentation. Regarding the Th17 cells, this is a very interesting cells. These cells, once it's a T helper cell, it's a CD4 T cells. Once they are activated, they and exposed to cytokines such as TGF-beta, IL-6, IL-23, and IL-1 beta. These cytokines induces, for example, these two transcription factors, our gamma T and STAT3, and these transcription factors keep constantly regulating the production of these cytokines, such as IL-17A, IL-17F, and IL-22. But in some situations, Th17 cells can also produce GM-CSF and TNF. And together, these cytokines is very important to maintain to promote the immune response in several infections, especially in bacterial infections and fungi infections, but also Th17 cells exert pathogenic function in autoimmunity, for example, in arthritis, psoriasis, multiple sclerosis. And one thing that is very important, like to any kind of cells, is that the T cells need to protect their genome against different stress that can harm their DNA. And it's very known, and we know that from the test book, like when we studied the T cells development, that during the recombination, the VTJ recombination, in order to acquire a TCR and a diverse TCR, during their development, the T cells need to undergo some DNA damage, but this is an example of a physiological DNA damage where enzymes such as RAG1 and RAG2 cuts the double-stranded DNA in order to occur the recombination. And later, there's a DNA repair pathway that acted to repair this DNA and therefore promote the proper development of these cells. So this is the most clear example of how, in this case, in a physiological way, when a DNA damage happens, and then in T cells in this case, and then it's repaired. But also when these cells become mature and go to the secondary lymphoid organs, they recognize the antigens, and here they go through several steps that needs to be particularly regulated to prevent DNA damage. As you know, after the activation, the T cells go through antigen to clonal expansion, and they proliferate massively. And during this proliferation, these cells can generate DNA replication stress, and this DNA replication stress needs to be repaired, otherwise the cells can enter in a cell cycle arrest, or can even, depending if it's not repaired, can even die, for example. But also another factor that can cause DNA damage here during this step is metabolic changes that happens during this process. As we know, especially effector cells, they are highly dependent on glycolysis to develop and then to differentiate. And during this process, sometimes the cells can generate harmful molecules such as reactive oxygen species, and also, as I'm going to talk in my next slide, ROS. In some situations, they can be it can be good for the cells, but also in high levels can be harmful to the cells. But also after these steps, these cells still need to go to the tissue where they will exert their response, and also in the tissue they can suffer several harmful things that can also lead to a genomic instability. For example, viral infections, bacterial infections, there are toxins that is released by these pathogens that can also cause damage in the DNA. And even though the inflammation that it's happening in that environment can also in some way cause some instability, genomic instability, and this can lead to impaired function of the cells. And regarding the reactive oxygen species, as I mentioned, ROS at some levels could be good to the cells, but at some in high levels can be dangerous to the cells. Here in the left panel, we can see an example where ROS is beneficial to the cells. Basically, after TCR and co-stimulation, the cells, of course, go through a metabolic reprogramming that leads to the production of small amounts of ROS. And these small amounts of ROS is actually good because they act in several proteins that is downstream the TCR and the co-stimulation signaling that later will stimulate the function of different transcription factors that acts in IL-2 production, for example, proliferation, survival, cytokine production. But in some situation, prolonged or strong TCR stimulation can increase this ROS generation, and increased ROS generation can therefore cause DNA damage, and this damage could come to some outcomes like cell cycle arrest, senescence, apoptosis, and more recently also something interesting that's been described in the past few years that in tumor, in cancer, for example, the tumor microenvironment can modulate the metabolism of even the tumor cells or the surrounded cells and cause, for example, metabolic changes that leads, for example, to accumulation of ROS and T-cell exhaustion. That's something that is very clear observed in tumor context, for example. And in the case of Th17 cells, ROS accumulation is associated with DNA damage. So this is an example of a study where they studied the function of this molecule called STIM1, which is a molecule involved in calcium signaling. And here in the red bar, as you can see, there's a CD40 cells with a mutation in this STIM1, and the cells that do not express the STIM1 accumulate more ROS generated by the mitochondria, and also these cells have more DNA damage, here accessed by the expression of DMA2AX, and this leads to cell death, but which suggests that in the absence of this molecule, Th17 cells is very susceptible to DNA damage, and especially to oxidative DNA damage. However, if you look at the blue bar here, the wild-type Th17 cells have mechanisms to protect themselves against this damage. So then our question was, how Th17 cells control this DNA damage? And this other study here shows that in a colitis model, when they isolate Th17 cells from the gut, for example, from the colon, from the small intestine, or lymphoid organs, these Th17 cells express genes related to this nucleotide excision repair. Of course, the focus of these studies was not this, but I think this is the advantage of the transcriptomic era that we live now. They found this information, and for us at the time was very important. And this nucleotide excision repair is one of the DNA repair pathways. that is involved in the repair of DNA damage caused by several factors. And one of the main important proteins involved in this pathway is a protein named XPC. And XPC is very important because it's the first protein to be recruited to the lesion site when the cells face a DNA damage. And once XPC goes to the lesion, you recognize this lesion. It leads to a signal transduction that recruit different DNA repair proteins that leads to the excision and repair of this lesion site. And thinking about all of these that I showed, we look at available RNA-seq data of in vitro differentiated Th17 cells. And we found that compared to naive CD4 cells, for example, Th17 cells upregulates genes related to this nucleotide excision repair pathway, such as XPC, XPA, and other genes. And basically, this leads us to the hypothesis that Th17 cells rely on this pathway to control DNA damage and maintain genomic stability. And this was a study that I did during my postdoc time in São Paulo with Professor Nils Olsen and Professor Carlos Wayne. And first, we use an in vitro model of Th17 differentiation. So basically took the naive CD4 cells from Y-type mouse and then put these cells in conditions that leads to the differentiation of Th17 cells. And then we compared the expression of XPC among different T helper subsets. So we see that Th17 cells compared to the others are the cells, are the subset that express most XPC. But also we see here naive CD4 cells express some levels of XPC. And then when we look at this confocal microscopy, we see that XPC and this other protein gamma2AX, which is a DNA damage marker, interact with each other in the nucleus of the cells, which suggests that during the differentiation, during their differentiation, there's a recruitment and an interaction between XPC and gamma2AX in the lesion site of the DNA damage. And then we did in vitro cultures now using XPC deficient Th17 cells. And we found that in the absence of XPC, Th17 cells do not differentiate proper. So they basically produce less IL-17A when compared to Y-type cells. They express less RR gamma T, which is the main transcription factor of Th17 cells. And also they have a lower expression of other molecules that is important to Th17 cells identity, such as TACH3, IL-23 receptor, and IL-1 receptor 1. So basically, this suggests that XPC is required for optimal Th17 cell differentiation. And then we want to know if this also happens in vivo. So we use a model of T-cell transfer colitis. Just a moment, the pointer is not working. Voltou. Okay, voltou. So we use, in order to understand if in the absence of XPC, Th17s also differentiate, how is the differentiation in vivo, we use this model of T-cell transfer colitis in REG1 mouse. So basically we isolate Y-type and XPC deficient Th17 cells and inject in REG1 knockout mouse. The Y-type cells, this is well known that they react against the microbiota and induce a chronic inflammation in the intestine. And then we evaluate how was the pode ser a bateria, né? Eu posso ficar aqui também. Você acha que ficar perto aí fica melhor? Não? So, então, Ele não está passando. Exato. So as you can see here, in the absence of XPC, the CD4, the XPC deficient CD4 T cells do not were not able to induce the intestinal inflammation. So the reg ones that received XPC deficient CD4 T cells have less signs of intestinal inflammation when compared to the ones that received Y type cells. And when you look at the mesenteric lymph nodes and the large intestinal lamina propria, we see that these CD4 T cells also produce less IL-17A. And something that was very interesting is that these XPC deficient CD4 T cells, when were transferred to the reg ones, besides do not produce IL-17, they start to express more FOXP3, which suggests that they are converting towards a regulatory phenotype. And then we want to explore the mechanism behind this. So basically, we did a bulk RNA-seq, and we saw that in the absence of XPC, Th17 cells have a downregulation of several genes related to DNA repair pathways, such as the nucleotide excision repair, but the base excision repair. And then this leads us to the hypothesis that that in the absence of XPC, Th17 cells was failing in having DNA repair to exert this function of DNA repair. So then our next question was, is this imperative function of Th17 cells in the absence of XPC due to more DNA damage once they are not expressing genes related to DNA repair? So then to answer this question, we measure gamma-2AX, which is one of the broad markers of DNA damage. And we saw that in the absence of XPC, Th17 cells accumulate more DNA damage. And we also evaluate this by a COMET assay that measure DNA damage, DNA strand breaks. And we also see that in the absence of XPC, there's more DNA strand breaks. And then finally, we want to understand what was the factor that was causing this DNA damage in Th17 cells. And we use a modified version of this COMET assay that it's by adding this enzyme called FPG, which is a DNA glycosylase that binds to oxidative purines in the DNA. When the cells have oxidative DNA damage, usually this enzyme binds there. So then we can infer if the cells are undergoing oxidative DNA damage. So basically, we saw that in the absence of XPC, there's more binding of FPG in this oxidative DNA purines, which suggests that there's more oxidative DNA damage in the cells. And then we look at the mitochondria of these Th17 cells, and we saw here both by FACS and by immunofluorescence that this mitochondria have more production or are producing more ROS. And this was associated with metabolic changing the cells. So there's an upregulation of genes related to stress response and downregulation of genes related to metabolic stress. And also when you look at the metabolism of the cells, the XPC deficient Th17 cells are less glycolytic, and glycolysis is very important to the differentiation, the function of these cells. They also undergo towards oxidative phosphorylation. And to make a long story short, what we observed in this paper was that during the differentiation of Th17 cells, signals like the TCR and cytokines induces the expression of XPC. This was something very interesting because when you look only in activated CD4 cells, only with TCR and co-stimulation without cytokines, if you remember that first graph that I showed, they express high levels of less levels of XPC, and I didn't show, but they express also high levels of gamma-2AX. So we basically, this third sign, that is the cytokines, seems to be the stimuli that is inducing this DNA repair machinery. And this is dependent on some transcription factors, such as BATF, RR-gamma-T, and IR4, something that I also didn't have time to show, but we also did some analysis on that. And then due to this production of ROS that is natural, something natural to the cells, XPC, probably together with antioxidant pathways, act together to control this damage that is caused during the differentiation of the cells, and this maintains the transcription program of Th17 cells. On the other hand, in the absence of XPC, the cells accumulate more DNA damage, and this DNA damage is related to increased mitochondrial ROS production. That is probably the factor that's causing this damage, these lesions in the DNA, and this impairs the transcription program of Th17 cells, making them produce less IL-17A and other factors that's important to their identity. And of course, these have opened a lot of possibilities to explore the role of XPC in Th17 cells, but also in other T cell subsets. But something that is very important is that Th17 cells, as I mentioned in my introduction, it's very important in different autoimmune disease. And one of the autoimmune disease that Th17 cells play an important role is the multiple sclerosis, and the experimental model, experimental autoimmune encephalomyelitis. In this disease, IL-17A disrupt the blood-brain barrier, which make the migration of Th17 to the CNS. And in the CNS, they produce IL-17A, GM-CSF, and this leads to an inflammation in the CNS that causes the demyelination of the neurons, leads to the signs of the disease in multiple sclerosis and in the EAE. And then we want to understand now if XPC contributes to Th17 cells mediate EAE development. And for this, Bruna, master student in my lab, is now studying through different approaches this question. We first are using a reanalysis of publicly available data sets from single-cell RNA-seq data to explore which cell types XPC is more expressing during EAE. We're also using the EAE model with MOG and CFA and pertussis to induce EAE in wild-type and XPC-deficient mouse, and to analyze how the Th17 cells differentiate in the absence of XPC. And first, we did some transcriptomic analysis from this data set from this paper published recently. In this paper, they isolate cells, both immune and non-immune cells, from the CNS of control and EAE mouse, and then they perform single-cell RNA sequencing. And then we download this data and reanalyze based on our questions to look how was the expression of XPC among different among different cell types, including these ones shown here in this UMAP. So this is a more broad analysis. So we want to first to understand if the expression of XPC is restricted to T cells, but of course not. So as you can see here, XPC and XPA and other factors are expressed in T cells, but also in resident cells from the CNS, like precursors of oligodendrocytes, in some other levels in other cells, for example, in the endothelium, but also other molecule, other genes involved in this pathway is expressed in the T cells, like p53, gamma-2AX, and other genes. But of course, our question is about the role of XPC in the T cells. So then we did a more deep analysis in this data set. So then we did we look at the clusters, the T cell clusters more deep, and as you can see here in the EAE mouse, we can see different clusters of T cells, including pathogenic Th17 cells and cycling cells, cycling T cells. That is the cells that migrate to the CNS and proliferate there when they arrive there. And then we run a gene score analysis that measure the expression of genes related to specific pathways. And as you can see here, both pathogenic Th17 cells and cycling T cells express genes more genes related to DNA repair and antioxidant pathways, for example, and downregulate genes related to oxidative stress, which suggests that the cells, when they arrive to the CNS, they kind of reprogram their gene expression to maintain a certain stability to exert their effector function. And when you look to the genes there, we see that the cycling T cells express genes related to the XPC pathway, this NER pathway, and also these genes are expressed in pathogenic Th17 cells, alongside with other genes such as p53 and A2AX. And then we want to test this hypothesis using a mouse model of EIE. So basically, we did the EIE using MOG and CFA and pertussis. So immunize the mice with MOG and CFA and then inject pertussis. This is a very classical model of EIE. And then we collect the cells from the CNS in the peak of the disease, and we look at and we stimulate the cells with MOG, which is the peptide from the myelin. And then what we saw is that XPC deficient mouse was completely protected from EIE development. It's a very strong phenotype, very interesting phenotype. And then when you look at the CNS, we see that there's no antigen-specific Th17 cells, Th1 cells, and pathogenic Th17 cells that co-express IL-17 and interferon gamma in the CNS. And this is a very interesting phenotype. Of course, this is a full knockout mouse. So you want now to generate a conditional knockout mouse for XPC. So then we can delete XPC specifically in different subsets, including CD4s. But until we have this mouse, we're gonna use some other approach. That is the passive EIE model. So basically, we're gonna immunize wild type and XPC deficient mouse, and then we collect their T cells and stimulate them in vitro and inject specifically this MOG specific Th17 cells. And then we can see if this phenotype is due to an intrinsic role of XPC in the T cell. And of course, we want to explore these mechanisms behind this in the XPC deficient Th17 cells. So now moving to the last part of my talk, I want to bring another aspect of this research line that I'm following the past few years. That is aging, and aging is highly related to persistent DNA damage. So we know very well that oxidative stress, replicative stress, and UV radiation are factors that cause genomic instability, accumulation of DNA damage, and this leads to senescence and aging. And a senescent cell that usually accumulates in the elderly is associated with increased expression of P53, P16, which leads to cell cycle arrest. Also with some alterations in lysosomal enzymes, and with production of a secretory phenotype that involves several pro-inflammatory cytokines. And if you go to the literature, we know that we can age differently, and this is related to our life habits throughout our life. So there's a physiological way to age, and this is more related to a health age, and is that old person that, for example, practice exercise, they eat well, they have like balanced diet, and this person have tumor suppression mechanisms, so they do not develop tumor, have wound healing, and have a better immune clearance or immune system. So basically, these are the ones that have good response in vaccines. They can deal with infections nicely. But on the other hand, we have another way to age that's a pathological way. And of course, there's several factors that can lead to that. But in this pathological age, one thing that is very interesting is that this is associated with chronic inflammation. And this leads to several disease that is observed in the elderly. And we are also interested in this aspect of aging, of how this inflammation influence different aspects of the immune system. And when it comes about the T cells, we know very well that during aging, there's a thymic involution, which reduces the output of naive CD4 cells, leading to increased memory cells. And when you look at the effector cells, effector T cells in the elderly usually have an exhausted phenotype, expressing inhibitor molecules, and some studies relate this to, for example, the development of cancer in these individuals. When you look at the cytotoxic T cells, these cells produce high levels of granzyme B, perforin, interferon gamma. So basically, in this pathological aging, the individual is inflamed. There's a chronic inflammation. But what about the regulatory T cells? The regulatory T cells is the cell that it's supposed to protect us against this inflammation. So it's the very important immune cells that maintain tissue repair, maintain the tolerance against microbial antigens and against the antigens derived from the diet. But what happens to the cells in aging, the elderly? One aspect of the Tregs that I forgot to mention is that the microbiota, the gut microbiota, it's a factor that is very important to the maintenance of these cells. So basically, the microbiota provides several factors such as short-chain fatty acids that's important to maintain the function of the cells and their function in maintaining tolerance, for example. But what happened in aging? In aging, it's well known that there's a gut dysbiosis. So basically, there's a shift between commensals to pathobionts, and this leads to increased intestinal permeability, and there's the translocation of these bacterial projects to the lamina propria, and this leads to remodeling of several immune cells in the lamina propria, which leads to a systemic inflammation with cytokines going to the bloodstream and going to different tissues of the body. And some studies in the literature already shows that regulatory T cells from the elder seems to not function properly. And basically, as I mentioned, in a young individual, the gut microbiota provides several factors that promote the differentiation, the function of these regulatory T cells. But in aging, this regulatory T cell seems to not function. So, and then our question was, is this loss of tolerance against this microbial factors that is altered during aging, the factor that causes this Treg instability during aging? So, so as I mentioned, there's dysbiosis, and regulatory T cells depends on this health microbiota to maintain their stability and their function. So then our question is this chronic inflammation in the elderly related to an impaired function of the regulatory T cells due to this loss of tolerance associated with this dysbiosis process? So then I start to do this project in Germany in the laboratory of Professor Adi Weismann, and then we start to explore this question. And at the time that I start this project, there was some evidence in the literature that when you give what we know already, that when you give old microbiota to a young host, this leads to T cell activation, and if you give young microbiota to an old host, these young microbiota are able to reduce this T cell activation. And then we ask if this could be due to, if this could be the gut microbiota of old mice be the factor involved in this impairment of the Treg function. And to answer this question, we did several experiments, but mostly we use young mouse that is 2 months old and old mouse that is 18 months old, so almost 1.5 years. And then we deplete the microbiota of this young mouse and give the microbiota of old mouse to this young through fecal microbiota transplantation. And we did the other way around. Like we deplete the microbiota of the old and give the microbiota of the young. And then we look at the T cells of this mouse to see what happened. And something very interesting is that if you look at the proliferation of these regulatory T cells here accessed by Ki67, when you look at the proliferation capacity of these regulatory T cells from the elderly, they do not seem to proliferate properly when compared to the young ones. Here in the red, when we deplete the microbiota from the old mouse and give the microbiota from a young mouse, this proliferation seems to be reestablished. So they start to proliferate again. And if you do the other way around, we deplete the microbiota from the young and give the microbiota from the old, the proliferation of the Tregs compared to the young mouse, it's impaired. So basically this microbiota from the old, it's causing some perturbation in the T cell homeostasis. And then we look at the effector cells, of course, and both CD4 and CD8 effector cells from the elderly produce more interferon gamma. We didn't see any differences regarding the IL-17, but this is something I already know. Aging have a strong interferon gamma phenotype. And then when you deplete the microbiota from the old and give the microbiota from the young, these effector cells produce less interferon gamma. And then to confirm this, we use a very nice tool that is germ-free mouse. And here we first start by looking at transcriptomic data from young SPF and age SPF mouse, and young germ-free and age germ-free mouse. And basically we download this data set from this paper, and then we focus on the T cell clusters. And what you see here is that the SPF, the mouse that was aged in a SPF condition, they have higher amounts of effector cells, but when they age under a germ-free condition without exposure to microbiota or any pathogen, they don't have these signs of effector cells. And we confirm this by FACS analysis. So when you look here in the SPF aged mouse, the both effectors, CD4 and CD8, produce high levels of interferon gamma, but when the mouse was aged in a germ-free condition, they do not produce lower levels of interferon gamma. And then we want to understand if this was because the regulatory T cells in this old germ-free mouse was preserving their function, their suppressive function. So in order to do that, we did that T cell transfer colitis model again. So we isolate naive CD4 T cells from young mouse and regulatory T cells from a mouse that was aged in an SPF condition and from a mouse that was aged in a germ-free condition and transferred to the regs. And what we observed was that here in orange, the regulatory T cells that isolated from the old germ-free mouse was able to suppress the intestinal inflammation when compared to the ones that came from a mouse aged in an SPF condition. And when you look at the proliferation capacity of the cells, these regulatory T cells from the old germ-free mouse are more proliferative and also able to suppress both Th1 and Th17 response. And then we want to understand what is the mechanism behind this, what is this microbiota causing to this, that it's causing this perturbation in Treg homeostasis in aging. And then we look at the single cell data again, and we saw here that the Tregs from old mouse housed in an SPF condition, they have more genes related to oxidative phosphorylation, ROS, apoptosis, TNF signaling, P53 pathway. And then when you look more closely to those genes, we found that the old regulatory T cells express genes related, upregulate genes related to DNA damage and senescence, and downregulate genes related to DNA repair, which suggests a sign of genomic instability in these old regulatory T cells. And next, we want to understand if this was because of the microbiota, so perform experiments to see if this genomic instability and senescence was dependent on the microbiota. And as I mentioned in the beginning of my introduction, the inflammation observed in the aging can cause this DNA damage. The mitochondrial dysfunction caused this through ROS, caused DNA damage and senescence, and this leads to this genomic instability. So basically, what we did next was to look at the ROS levels in this regulatory T cells. Here in black is a young Treg, and here in green is an old Treg. And as you can see here, the old Treg have more ROS intracellular. Here in red, it's an old mouse that we give the young microbiota, and then we can see here that this Treg now have less ROS when they got in contact with the young microbiota. And here in blue is a Treg from a young mouse that now received the old microbiota. So basically, this old microbiota, it's increasing in some way the ROS levels inside the cells. And ROS, as I mentioned, is one of these factors of DNA damage. And then when you look at the germ-free mouse, we observe that the old germ-free mouse have less signs of DNA damage and senescence, here measured by gamma-2AX and P16 when compared to the old SPF mouse. And then to finish, we want to understand which factors was causing this genomic instability in this old regulatory T cells that was driven by the microbiota. And as you can see here in our single cell data, TNF signaling pathway was one of the pathway that was upregulated in these aged Tregs, and TNF signaling is detrimental to the Tregs. So basically, the TNF is toxic to the Tregs if this TNF signaling through the TNF receptor 1. And then we measure the TNF receptor 1 in the young and old regulatory T cells. And as you can see here, mesenteric lymphonodes and in lamina propria, the Tregs from old mouse express high levels of TNF receptor. And this is followed by increased levels of TNF, both in the colon but also systemically. And then when you give the young microbiota to an old mouse here in red, these TNF levels are reduced. So then we next want to understand if this TNF signaling in these regulatory T cells was the factor that was causing this instability in this Treg. So basically, unfortunately, we didn't have an old TNF receptor knockout, because then we need to wait 2 years, almost 2 years, to get these knockouts old. So then we use an approach that was use young Y-type mouse and young TNF receptor 1 mouse, which we deplete the microbiota and give the old microbiota. If you remember from my previous results, we observed that when you give old microbiota to a young Y-type mouse, they got more interferon gamma. But when you give this old microbiota to a young TNF receptor knockout mouse, they didn't get this high levels of interferon gamma. And when you look at the Tregs of this mouse, we saw that when the Tregs from young TNF receptor 1 got in contact with the old microbiota, they express less signs of senescence, DNA damage and senescence, here through the gamma-2AX and P16. And now they start to proliferate again. So they increased their proliferation when compared to the Y-type Tregs that's in contact with the old microbiota. So basically, this aging microbiota is possibly promoting this TNF signaling because of this dysbiosis. And this TNF, and when these old Tregs are exposed to this TNF signaling, they got more unstable. And finally, to confirm that in a Treg specific way, we use a system to try to reach to a Treg intrinsic way of TNF, of deletion of the TNF receptor 1. So basically, we use Y-type young Treg and TNF receptor 1 young Treg that was distinguished by congenital markers. And then we inject these Tregs in an old environment. And then we let these cells there for quite a few days to see how they behave. And then we saw that when the TNF receptor 1 deficient Treg are in contact with the host environment that, as I showed before, is full of TNF, they do not, they express less gamma-2AX and less P16, which suggests that this environment full of TNF is what's driving this genomic instability and this senescence phenotype that we observe in this ultrasound. And to make a long story short, again, we observe in this study that this dysbiosis caused by this during aging leads to a shift in different bacteria that I didn't have time to show, but this dysbiosis leads to the production of high levels of TNF, not only TNF, but other cytokines that we didn't explore, but of course is increased there. And these regulatory T cells from the elderly are sensible to this highly inflammatory environment, and they become unstable and they produce more oxidative stress, which is associated with increase of senescence markers, DNA damage, and less proliferation, which is also a sign of senescence. So with that, I would like to thank all the people that was involved in both of these studies in São Paulo, in Germany, also the founding agency, and also now all the people here in Rio that it's also helping through this last year, especially my students, also people in the Department of Immunology, my department, also in the Institute of Biomedical Science. Thanks to Flavia Alcantara Gomes, I'm able to continue this aging research now because she is one of the experts in aging research here in Rio, and she developed a super nice mice facility to age mice. So then I'm continuing now this research line, and of course, all the financial support here. So thank you so much for the attention.
All right, it was a great talk. We'd like first to open the floor for questions from the students, if we have any. Do we have any questions from São Paulo, from the students in São Paulo?
Bruno, not from students. I have a couple of questions, but let's wait. Students in Rio or online.
I'm sorry, Gustavo, we missed what you said.
I said that there's no question from students.
No questions? Okay,
not here. I will have a question, but you can start.
All right. We are going to, we have a few questions here. Question from Gustavo. We are going to start with Miriam.
Well, first of all, thank you very much, Jefferson. It's beautiful to see all the research that you've been doing and this building up at the lab. I have actually, like, it's more for a thinking. One practical question is, have you tried co-housing the aged and the young mice and see if they level up in the middle, or if one brings the other? That could be like a co-housing of humans, maybe something like that.
Yes, this actually was the first experiment that I did when I started this aging project in Germany, because it's much easier. And then I remember at the time, because my hypothesis was different, was that when I let me remember now, when I co-housed the young and the old, at the beginning, my idea was that the old phenotype would be transferred, like the old microbiota would be transferred to the young, but was the other way around. Like the young transferred the microbiota to the old, and then the old start to make less interferon gamma, and the Tregs was function better. And then, yeah, we did that, and this is the outcome. But this is because I think this microbiota from the old is so, sorry for that term, fucked up, because they are really altered. So, and then, yeah, that's why the young microbiota are more prevalent. Then it's able to survive there and repopulate there.
Right, so I have a question from Marcelo Boza, if you could come up here so people in São Paulo could also see you.
Hi, congratulations, was great talk. So I have a few questions. One, related to the colitis model. You observed that the Th17 was reduced and you have an expansion of T regulatory cells. In the EAE model, you observe again almost a total absence of Th17 and also absence of a Th1 producing a gamma interferon cells. So in the EAE model, do you see also an expansion of the regulatory T cells? Would that be the reason that you have almost a total tolerance in this model? And is this effect of XPC deficiency related to an intrinsic problem in a T cells and a benefit to regulatory T cells? So that would be the first question. And the second one would be related to the aging. So how stable would be this microbiota after transfer? So my question is, can you build up these microbiota and they become stable in the aged mice? Or the aging environment would be turning these microbiota over and over again?
Thanks, Marcelo, for the question. Regarding the first question, yeah, we see more Tregs in the EAE, also in the absence of XPC, but this is something interesting because at the steady state we already see this. Like the XPC mice have more regulatory T cells, and these regulatory T cells are more activated. So this is something really intrinsic that we don't know yet because it's a full knockout. So we don't know if this comes from the thymos or from myeloid cells. This we don't know yet, but yes, it seems that XPC is preferentially expressed in the effector compartment and in some way restrains the regulatory compartment, but this we don't know. We have no idea yet. So this we're gonna figure out when you do a sequencing of these cells in the future, so that we don't know exactly. And regarding your second question, yeah, I mean, I didn't follow longer. Like usually this protocol, this fecal microbiota transplantation protocol, it's 15 days after you give the microbiota, then we open the mouse of 15 days. But yeah, I believe that it is stable because yeah, after 15 days, the young microbiota was already able to make that environment anti-inflammatory again. So yeah, but I don't have data to show that, unfortunately. But it's a very good question because yeah, maybe becoming stable and becoming inflamed again. But yeah, the only thing that I can say at this point, 15 days, this microbiota, it's making the environment anti-inflammatory again. But it's interesting. Thanks.
Right. So now we have a question from Gustavo.
Hi, Jeff. Wonderful talks.
Thanks.
Two great piece of work. Marcelo actually stole my questions. One was you already answered, whether the aging microenvironment would change back. So how is this balance? I think is still unsolved, according to your question. And the other is regarding the different subsets of the T helper cells. Why XPC operates in this way in the Th17 and not in the others, particularly the Tregs, because the Tregs actually makes a better compartment in the absence of XPC, and why a lack of DNA repair would interfere in the EAE. Actually, that's the question.
Gustavo, can you repeat the last part of this last question? Because
the last part is why the lack of DNA repair, okay, the XPC deficiency, it's is a deficiency in the DNA repair. Why this would benefit actually, the EAE model?
Okay. Answering your first question, I guess was like why XPC specifically expressed in Th17 cells, right?
Yes.
Well, this is a question that a lot of people already asked it. Yeah, we did this in vitro. So in vitro, you know, it's all beautiful. You put that soup of cytokines there, and it's easy to make this environment. So yeah, this data is in vitro, and we saw that it's specific in Th17 cells. But if you look at the literature, different T helper subsets deals with ROS in different ways. For example, Th2, for example, yeah, they seems to like high ROS levels to become better differentiated. But when you look at the knockout mouse, like in this in vitro setting, we only saw differences in Th17 and Treg. So it seems that this function of XPC is more specifically to Th17 and Treg compartment. But yeah, I assume that why Th17, not others, because yeah, the cytokines and the transcription program leads to this pathway, not other pathways. But yeah, there's other pathways also that is related to this DNA repair. And yeah, we want to evaluate this in the future. Like for example, the base excision repair, it's a pathway, is actually the canonical pathway that is involved in the repair of oxidative damage, and probably is also working Th17 cells. But I think that it's not something specific of the Th17 cells. So I think it's very, all of these pathways, it's working in different ways together to maintain the genomic stability of the cells, because they are highly proliferative and they deal with this lots of ROS. And your second question about the EIE, I guess is a consequence. Of course, when only gonna know about that when you do the passive EIE, specifically looking specifically the function of XPC in CD40 cells. But I think it's a consequence. Like if you already give a cell that is producing less IL-17 and express more FOXP3, I think I'm already creating tolerogenic environment there that it's yeah, preventing already these cells to become effector cells in the case of EIE.
All right, so you think that in the XPC mice, you have less Th17 differentiation, and when you do have it, these cells are damaged and then they die.
Exactly.
Okay.
I don't think they die. That's
They
the
go
next.
senescence. Maybe they go senescence.
Yeah, because I didn't show that, but we measure cell death in this in vitro model, also in vivo, and we actually never saw more apoptotic cells, for example. Didn't measure other cell death types, but at least they are not dying, as far as you know. So I think this is, as they have all of this damage, they need to deal with that, and the way that they are dealing is going through a cell cycle arrest and maybe becoming senescent. But this is the next step that I want to investigate now.
Okay, thank you very much. Anything, just to finalize, anything on the CD8 compartment? And
Yeah, that's actually my next step. I also have a student now that is looking the CD8. The CD8, what we have so far is that when you restimulate the CD8s in vitro with anti-CD3 and CD28, they produce less interferon gamma. They do not proliferate properly. So I think it's a similar mechanism as in the CD4s.
The XPC knockout cells.
The XPC, exactly.
Thank you so much, man. Congratulations.
Thank you, Gustavo.
Right. So I have a question now. I was wondering, yeah, I don't know if I missed something, but it seemed a little bit paradoxical. When you look at the single cell reanalysis data, you could not see any increase of XPC in the Th17s, right? And if you would start with that data, I guess you would never look at XPC. But when you do the actual disease model, the XPC deficiency really gives this beautiful phenotype. So what do you think it's going on? And we are going to lose the computer now.
So
Okay. We are back here over the phone. We have some problem here with the no break. I just realized now that the no break was not working properly.
Yeah.
But what's going on now? So you don't need this anymore? It's not working.
Okay.
What do you think is going on there? It's that XPC is working at different place than what's the single cells are coming from.
Yeah, it was very interesting because OPCs was expressing high levels of
Right.
XPC. And I think, of course, as I mentioned in the beginning, the inflammation could drive this DNA damage. So probably, yeah, this was the idea at the beginning of this reanalysis of the single cell to see during the EAE is a specific, this is an expression of XPC's T cell specific or is also expressed in other type of cells. Probably there's other cells. Also, we saw, I didn't show, but myeloid cells express a lot of XPC. So I think this would be the advantage of having a conditional knockout mouse in the future, because then we can delete specifically, see what's the most important cell type there that is suffering from this DNA damage that it's causing the disease, and how this is. Nopsis is related to the remyelination of the neuron. So
yeah, but it could also not be explained by the participation of XPC at the sensitization phase of the model, and then when you do the single cell, you only do on these cells that are actually infiltrating. You actually miss this initial part.
I think this is related to Gustavo's question. Yeah, we didn't look at deep in the priming phase in the lymph nodes, for example. But yeah, as we already start in an environment that it's full of Tregs in the XPC mouse, so probably this Tregs is suppressing and it's not already leading to a proper Th17 development. And together with this phenotype that we already observed in vitro, they are not generating properly. Yeah, and also these Th17 cells, like in the colitis model that I showed, these Th17 cells, they already, they are very plastic cells, so they is converting to Tregs. So besides the Tregs they already have there, when you give the antigen co-stimulation and the cytokines, because of TGF-beta, of course, this XPC deficient Th17 cells is expressing FOXP3. So probably, like I say, maybe already in the priming phase in the lymph nodes, that could be also a factor that is protecting the IE.
Bruno, we have two.
And just one quick
questions
question to follow up on that, because I was gonna ask you if you thought that in the aging animals you had a skewing towards the TCR. So I was gonna ask if you had looked at TCR usage, basically sequencing the TCR of those cells. But since when you do the exchange, you do get lower TNF again and all that, then it doesn't seem to be something on that end. But since you mentioned now that it could be selecting clones specifically, are you thinking of looking at that?
Yes, in the future, yes, like this, especially because one thing that's very interesting in this aging model is that the Tregs accumulate. Besides, they do not proliferate when they are restimulate, they accumulate. If you already take a 1-year-old mouse, they already have more Tregs when compared to a young mouse. So I think that probably there's something related to this microbiota that's making these cells expanding, but at some point they lose their function. But yeah, I intend to do this in the future.
Okay, let me see, because we had a couple of questions here. Let me see.
We also have a couple of questions from students here.
Maria,
Você falou, desculpa, você me chamou? Posso fazer minha pergunta?
Miriam, are you hearing me?
Yeah, Maria, please go ahead.
Oi. So very nice, wonderful work, Jefferson. Congratulations. Yeah, some of the questions were already answered, but I still have one. which is about the Tregs in the germ-free mouse. When you compare the old and aged germ-free mice, there were some differences between them. So my question is not so how can I say, evident like in the SPF mice, but you do see something. So do you think this intrinsic difference in T cells, aged T cells, could be one of the reasons for the dysbiosis, and this because it is a two-way route. I mean, one interferes on the other. So I would like to have your view about that. If there is something which is intrinsic to aging in T cells that can contribute to the all scenario you're seeing.
Maria, thanks for this question. Actually, this is the $1 million question because we don't know who came first in this case. If it's this dysbiosis that's causing this inflammation in the aging, or is the aging that's causing the this dysbiosis? I mean, I think it's the aging environment some way at some point, of course, not already at the end when you already get old, but during their life span, there's something that anyone knows. If you go to the literature, you don't know exactly what is this factor that is triggering this inflammation. And then, of course, this inflammatory environment is causing this dysbiosis. And of course, when these Tregs get there, they need to suppress this inflammation. They do not get that metabolites and factors that it's important to their function, then they cannot function properly. So yeah, and this in mouse, it's quite easy to think, but in humans, for example, it's completely different because when you like throughout our life, we eat different diets, we use antibiotics, several other factors that can change this microbiome. So I think in humans could be different, but yeah, this is something that we don't know yet. What it's really causing this dysbiosis if it's the aging or the diet. I don't know if I answer your question.
Yes, thank you so much. Congratulations again.
We had, well, not a question yet, but someone that would like to ask a question, Maria Fernanda Yamashita Barbosa. There was another name, I believe it's Costa. Maria Fernanda, would you like to ask a question? Write it up on the chat. Let's see. Okay. MSDC have the same situation of Treg?
Honestly, I don't know. Maria Fernanda, né?
Being affected by TNF, yeah.
Yeah, honestly, I don't know, Maria Fernanda. It's it's not the literature. I don't follow this literature about these MDSCs. Yeah, because it's myeloid compartment, and yeah, I don't know. Honestly, I don't know. Sorry.
Bruno.
Yeah. Yeah, go ahead.
We have two questions here from students.
Oh, sure, please.
Hi, Jefferson. Really nice talk. Thank you.
Hi.
Do you hear?
Yeah.
So you saw lower STAT3 phosphorylation on XPC knockout mice. We are wondering if you have tested if inducing STAT3 activation could recover Th17 function and differentiation.
Okay, this is a very interesting question. Actually, didn't have time to do this experiment, but yeah, I mean, honestly, I think that will not recover because the cells is already facing this DNA damage that's impairing their transcription program. So I think if I put more IL-6 there, I would be forcing even more the cells to do something that they are already not doing. Do you understand? But what we saw is that if you remove this, the causes of this DNA damage, that it's adding, for example, NHC2-cysteine, that is an antioxidant molecule, we can restore the IL-17 production, we can restore the phosphorylation of STAT3 and gamma T, for example. But the problem is that it's what is already happening in the cell, like this oxidative DNA damage.
Thank you.
There's another one here. Henry.
Thank you. So Jefferson, thank you for your talk. It was very interesting. My question is very general. We know that CD4 are very plastic.
Sorry, I cannot.
Okay. Okay. You listen now? Hello. Hello. Hello.
Pode falar.
Okay. We know that CD4 are very plastic in generation and changing their immunophenotype. Do you consider this aspect or this?
I'm sorry, but you have to speak a little bit louder. We're having trouble to hear you here. Yeah.
Okay.
Yeah.
My question is about the plasticity of CD4. Could I know if you consider this aspect, if the microbiota can impact in the plasticity of CD4? And the other question is if you explore that innate cells that has this phenotype TC17. Thank you.
Yes. Sorry. The first question, just to audience here, the first question is about the plasticity of CD4 T cells. I'm not sure if I understand properly the question. Yeah, in the XPC context, we saw that XPC, as I mentioned before, is more specifically a factor that regulates TH17 and regulatory T cells. The other cell types, TH1, TH2, we didn't saw differences in their differentiation and function. They seem to differentiate and function similar in the absence of XPC. And the second question is regarding if we saw this process, the same process in the innate cells. Now I don't know what innate cells you are referring to. I mean, we didn't look at myeloid cells. We didn't look at any kind of myeloid cells, and also we didn't look at innate lymphoid cells. So it's something that I want to do in the future. Look at the ILC3, for example, that it's like an innate TH17 cell, but yeah, we don't have data on that, so we don't know.
Okay, so I guess we conclude the questions that we had today. We thank very much for everybody that was in the audience, and hope to see you guys again next week for another installment of the webinars. And thank you very much, Jefferson, for a really exciting talk, very interesting data. I'm really looking forward for the next papers that will come. All right. Thank you very much. Bye-bye.